Okay, hi everyone. Okay, so let's see. If you guys have any questions, go ahead and say hi. Oh, hi, Mr. Chow. Thank you. Okay. Mr. Chow wanted me to wear a costume. I don't have a costume, so this is the best we're going to do. All right. This hat is very big. Okay, so as a quick reminder for the AP exam, there's Mr. Chow. Hi, Mr. Chow. So I know there was some confusion as to how many free response questions you were going to have. So I looked it up, or multiple choice and whatnot. It looks like based on the AP exam website that you should expect 63 multiple choice and then 6 grid-in questions and then 8 free response, 2 long, 6 short. That's at least the official word on their website. I know that the practice test you guys took was not quite that, at least in terms of multiple choice. So just kind of know what to expect, I guess. I'm not sure why that practice test is shorter. Maybe they took questions out after the fact. I'm not sure. This hat is very big, so I'm going to take it off. All right. So do you guys have any specific questions? I know some of you likely went to the review today, and so you might then have a lot of stuff answered there. Oh, AP Week 11 for my students. I'm sorry. I will upload it right after this. I will remember to do that. Okay, let's see. What can I tell you since no one's asking me questions yet? So after you've taken the multiple choice, you probably noticed that they're not necessarily asking you for like a lot of like specific details, which is a little different, I know, than some of the practice questions we had you do for homework, because the ones we had before were really more like before they did a redesign in 2013. So just know that no matter what, you want to approach the multiple choice sort of like process of elimination. You want to go through each answer choice and really kind of think about like what could be wrong about this question, what could be wrong about this response in the context of the question, right? Yes, totally. I can go over cyclic and non-cyclic electron flow and photosynthesis. So remember that for the light reactions, and yes, I will get the Krebs cycle stuff in a second. Okay, so for cyclic and non-cyclic electron flow, in both cases, it's the idea that in the thylakoid discs of the chloroplasts, you have these electron transport chains, right? So in non-cyclic flow, which was what we covered first, that sort of, I guess you could quote call the standard view, kind of photosynthesis, right? Which is to say the light hits the two photosystems. Remember, they're in not opposite order, but they're in different order such that they're not going to be, sorry, Mr. Chow is too big. It's falling off my head. So it goes photosystem 2, then photosystem 1. And so the idea is that the photons of light will hit those electrons at the center of the photosystem and excite them. And then those electrons get passed down electron transport chain. So the electron from photosystem 2 ends up, after going around the electron transport chain, it makes ATP and ends up in the photosystem 1, right? Photosystem 1's electron gets excited, go down the electron transport chain, ultimately goes to NADP plus to make NADPH. So in non-cyclic electron flow is when you are going to produce both ATP and NADPH. Whereas in cyclic electron flow, the idea is that if you are using up ATP faster than you are using up NADPH, then you don't necessarily need to do all of that, right? You could just essentially excite the electron in photosystem 1, which is the second one, have it go down the first electron transport chain and land back in its original seat. So you're not having to also replenish that electron. And in doing so, you're only going to generate ATP. Plants typically can switch between cyclic and non-cyclic flow then, depending on what their needs are. If they need both ATP and NADPH, or if they only need ATP. Okay, what happens to the citric acid after it's produced during the Krebs cycle? So citric acid, it's also the name for this Krebs cycle, right? So what you want to remember the Krebs or citric acid cycle is it's a cycle, meaning you not only have to do what is sort of the main idea energy-wise, which is making NADH and FADH2, right? But in the process, you also have to regenerate that 4-carbon compound that's going to come in, because acetyl-CoA is coming into the citric acid cycle, and it's joining with a 4-carbon compound, right? So when it does, it's going to go through all those different transitions, and in the process generate NADH and FADH2. But if you want the Krebs cycle or citric acid cycle to restart, right, then you have to regenerate that 4-carbon compound. So essentially what happens in the citric acid cycle with citric acid, without going to like exhaustive detail, because I don't think the AP actually needs you to know that, is that the citric acid cycle is going to have all these different enzymes, right? And so they're going to slowly take that citric acid and convert it into different intermediates, ultimately losing some CO2, right? And that's where you end up back with the 4-carbon compound that is now ready to receive more CO2 and go through the cycle all over again. I think if you know it sort of just to that extent, you're fine. Like they definitely are not going to ask you to memorize or show them what you know specifically about the citric acid cycle, past sort of the big idea of it. Okay, Manish says, right, I mean, number one, you can judge from the FRQs, like how much space they give you, right? So with the long questions, which on the practice test we did was number one and number two, they give like almost 2 pages to write. So they do expect a more detailed response, right? And typically those ones are going to have more parts to them. Either way, though, keep in mind like question one, question two, right? They had like 4 parts, I want to say. Each section is only worth like anywhere from 2 to 4 points. So you also want to be careful not to like spend too much time on them, right? Because each section essentially is only going to yield you 3 to 4 points. That being said, you want to make sure you answer their question. But yeah, I would say that then with a short one, right? I mean, that one total is only worth 3 or 4 points. So definitely, it's not about the length, but I think from a timing perspective and how much time you're spending on it, you want to make sure that you're not spending too much time on it, right? I mean, if it happens to be that you write a lot, I don't think it's the end of the world if you're doing it in this shorter period of time. But if you're writing as much as you do for the long ones and you're spending like 20 minutes on a short one, then that's definitely too much. Because remember, you're going to have 10 minutes to write, 10 minutes to read, and then you'll have 80 minutes to write. So 80 minutes to write for 8 questions. I mean, you could divide it up into like 10 minutes a question, but you they're expecting that you're going to spend longer on the long ones, right? The AP exam for 2018. Let me look that up for a second, because to be honest, I haven't looked at it. So, okay, thank you. Let me see if I can click on that for a second. And yes, I can review how CAP and CAMP work. So while I try to open this up on my computer, because it is a little bit unpredictable. Okay, let's see. Okay, so let's talk about CAP and CAMP while I look this one up. So CAP and CAMP are part of what we would call positive gene regulation in operons, the lac operon specifically. So the idea is that they CAP and CAMP work together, right? So you're with the lac operon, you're always going to have CAP around, but CAP is not able to bind to the promoter region when CAMP is not around, right? So essentially now it's like they're the cheerleader and the pom-poms. That's why I say where like one essentially changes the shape of another, so that now it's more likely to bind there, and that just causes the RNA polymerase to be more attracted to that area, so that transcription is likely to happen faster. That's sort of a quickie version of the explanation, I guess. And so really it's a good example of positive gene regulation, because a lot of time we talk about negative gene regulation, how we shut down a gene and things like that. But this is in ProCare, it's a very specific example of how you would make the operon transcribe faster or transcribe more than it normally would. Okay, let's take a look. How do we build the cladogram from question 1? Okay, so let me read the question. Let's see what we've got here. Polar bears, okay. 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 Let's see. Construction. Oh, okay. So let's see. So for this one, right, you have the panda, the black bear, the brown bear, and the polar bear. So it looks like what they want you to kind of look is that sort of where you do find the most differences between them, right? So it looks like offhand the panda has the most differences with everybody. So I think the idea would be like when you draw your cladogram down here. So you can see like so they have the, like, I think on, what is it, the second page, right after the table where they say like construct a cladogram. I would imagine that you would then put the panda on the first branch, the one that branches off the first, because it has the greatest number of differences. And that would be your outgroup, right? Because it's the most different. And then from there, you also have the brown bear, the black bear, and the polar bear. So when I look, I see that let me see what the question's asking again. Yeah, okay. So then if I look at black and brown bears, right, then I see the fewest difference between them, because there's only one amino acid difference between them. So I'd put them all the way on the right with the 2 branches, right, which would then leave the polar bear, which is kind of in the middle, because the polar bear has essentially 7 amino acids different from the black bear and 8 from the brown bear. So I guess if I was going to do that, I would probably put brown all the way to the right, and then black, and then polar, right? Brown, black, yeah, polar, and then panda. That would be my guess as to how they want you to build it, because they want you to look at the amino acid differences, and based on that, assuming that amino acids would be more similar amongst organisms who branched off later from one another, then you would, put the ones that are more similar together. Okay. Thank you for okay, I think I got the link then, yeah? Okay. Does that help, hopefully? I mean, they don't have an answer, so that's my guess as to how I would build it. Okay, other questions you guys have? Oops, sorry, I'm just getting a message. Like on the nodes. Okay, but why couldn't you? Oh, okay. Let me see the picture. Yeah, I mean, I honestly don't. I think, yeah. I mean, I'm honestly not sure if I don't know that it would really matter if you switched it. I guess the only reason I would be inclined to put what is it, the black bear, what is it? The black bear closer to the polar bear, just because there's 7 versus 8 8 amino acid differences, but I don't know honestly that it would, make that big of a difference based on the way that they drew their tree, right? I mean, it's probably not that different. And maybe they'd accept both, I don't know. I mean, you know. Okay, for the same 2018 test, can I explain 2C? This is number 8. Oh, okay, hold on. Okay, so let me see what you sent me. Let me see if I see 2C on here. If I don't, I will. Yeah, that looks like it was only 2C. Well, it's 11 pages long. So let me see. Or is it sample answers? Yeah. Okay, so give me a second then. Okay, so Erica, you want 2C? Yeah, let me take that look at that. Oh, and then 2017. Do we have? Oh, okay. Hold on. I don't have the 27 FRQs, so you guys are better at finding things than I am. Let me look up 2C first, and then I'll try to look at the estrogen thingy. But I'm not sure what that is either. Okay, let's see. Let's see. That's 2C. Oh, this is question 2, right? Okay. Is this question 2? No, this is still question 1. I'm sorry. Trying to find it all. Okay, 1. Okay. Yes, this is 2. Okay. Okay, let's see. Sorry, I just have to read the questions very long. Okay, very quick. Okay. Okay, a student claims that the NFKB protein is located in some person. Let's see. Okay, so they want to know. Oh, sorry, I lost this here. 2 fractions where internal. Okay. Until the protein is needed for transcription. Justify the student's claim with evidence. Identify 2 fractions. Huh. Okay. Let's see. Well, I mean, when they say identify, I assume they mean just saying fractions 2 and 3. I think what they want. I'm imagining what they want you to tell them is to say something like, well, if the student's claim is that it's located in the cytoplasm until the protein is needed, that you would say that if this was the case, then fraction 2 and fraction 3 would be sections of the cytoplasm, maybe. Identify 2 fractions where N-terminal, it's N-terminal gastrin. This is that enzyme, right? Oh, okay, I see. It says when. Oh, there's a picture. Sorry, I missed that. Oh, now I see. Okay. Yeah, I mean, I think they just want you to sort of tell them based on this diagram, like where you would expect to see this cell. I think they want you to show that you can read the graph and show that, you know, it would be in the cytoplasm, that you wouldn't find it elsewhere. I think also because they mentioned that it's located in the cytoplasm until the protein's needed for transcription. Maybe you could also argue that you would find it in the nucleus once it is you know, could find it maybe in the nucleus once you have active amounts of like RNA polymerase or something like that. Let's see. Oh, okay, Audrey, hold on. Let me look at that one now. And then, oh, there was a question about something, equilibrium, I think I saw. Okay, give me a second here. Okay, so yes, we will go over chi-squared in a minute. Thank you for reminding me, Tajin. Okay, this is number 8. Why would antibodies be ineffective in treating estrogen-dependent cancers? Okay, let me take a look at that one then. I'm not sure which one it is anymore, so give me a second. It must be this one. Okay, so number 8. Okay, hold on. Nope, only 6. Okay, 7. Okay, estrogens. Okay. Okay, they generate antibodies that bind to purifying estrogen receptors extract from cells. Oh, okay. I think in this case, the key is for you to notice that they tell you that estrogens passively diffuse across the plasma membrane and bind to their receptor proteins in the cytoplasm of target cells, right? So, in that sense, I could see how if you have these antibodies, which are proteins, right, they're not really going to be able to prevent, they can't get across the cell membrane, right? I mean, not without some sort. Typically, like antibodies and proteins, that they're going to bind on the outside of a cell. So I would imagine that the key here is for you to notice that they're small, they're hydrophobic, the estrogen, right? So if the antibody is supposed to somehow block the estrogen from binding and now stop the cancer from doing something, it's not really going to work, right? Because the estrogen doesn't need anything to cross the membrane. So it's still going to be able to cross the membrane, bind to their receptor protein on the inside of the cytoplasm of the target cell. But your antibodies don't have that same liberty. Like the antibodies can't cross the membrane. So they're going to be on the outside. So the reason that they generate the antibodies is, notice that they said to purified estrogen receptors extracted from cells. Like they did it in a lab, right? In a petri dish where they pulled out these receptors and then saw that, oh yeah, the antibodies can bind. But when they do it in an actual cell, it doesn't work because the estrogen just, the receptors on the inside in the cytoplasm. So the estrogen goes over by itself and diffuses across the membrane because it's small and hydrophobic. And then the antibodies can't do the same thing. So they're not really going to be able to block it because they're on the outside and the estrogen can get inside by itself. And that would be how I would answer the question. Okay, so let's go chi-squared first, and then we'll do coupled reactions. And then Manish, I'll get your question on taxonomy. Okay. Oh, 8B. Oh, sorry, did I read the wrong question or did I get that one? Let me see. Let's see. Yeah, okay, no, I got 8B. Okay. Okay, so chi-squared and Hardy Weinberg equilibrium. So remember, I mean, they basically want you to Okay, so first of all, they may or may not ask you chi-squared. I know that a lot of years they have, but I want to say 1 or 2 years ago, someone came and said they didn't ask a single chi-squared question. So keep in mind they might not ask you. With chi-squared, you want to remember that they will give you the equation, right? They do not expect you to memorize the equation. But what they do want to see is, can you figure out how to use it? Now, we use it in the context of Hardy-Weinberg. Also keep in mind, it doesn't have to be Hardy-Weinberg. If you go and look up the 2013 test, it's online somewhere. I think question 1 has to do with like putting bananas in different parts of an experimental apparatus and like seeing if the flies go there or not, right? And in the end, they want you to do like a chi-squared analysis based on what you call a null hypothesis, right? Which is that there will be no effect if you don't, if you put ripe banana on one hand side or you don't. So just keep in mind that chi-squared is anytime I want to test what I thought I was going to get with what I actually got. And so you always have your observed, which is what you actually got, versus your expected, which is what you thought you were going to get. With Hardy-Weinberg and genetics and things like that, it's based on probability and Punnett squares, right? So in any situation, you want to be able to say, like, well, if I'm doing chi-squared, what did I think I was going to get, or what is theoretically what I should get with what I actually got? In which case, then you do observed minus expected squared over expected, and you would do it for as many categories as you have, right? So if we're talking about Hardy-Weinberg and we're talking about like 2 different genotypes, then it would be like what I thought I was going to get with the Punnett squares compared to what I actually got as my data. Also remember with chi-squared, you have to make sure you're doing chi-squared with numbers of organisms and not percentages. And what I mean by that is be really careful that you're not like, oh, I was expecting, genotype frequency of 0.7. Because if you're using a number under 1, it's always going to end up really small, right? Whereas instead you want to say like, well, if I was expecting like 75% of 100, then it's going to be 70 flies instead of like 0.7. So make sure you're always doing your chi-squared with numbers of individuals, not percentages or frequencies. With Hardy-Weinberg then, right, if you're doing chi-squared there, it's the idea of like what you thought you were going to get, what you actually got. So observed is what you actually got, and expected means your Hardy-Weinberg numbers, which is where you do p squared, 2pq, and q squared, right? Where p squared is the frequency of the what is it called? The homozygous dominant. 2pq is your heterozygotes, and then q squared is your recessives. So in that case, again, though, you want to be careful, right? Because that automatically lends itself towards using percentages. So if you're like, oh, my Hardy-Weinberg says I should have gotten 50% big G, then you're going to want to figure out, well, in the actual population of like 200, right? Like what's 50% of 200? Not using 0.5, but rather you would use 50% of 200, which would be 100. Sorry, Tajin, does that help? I know I didn't actually give you a specific problem, but if you want me to, I can pick one out. Oh, which, like, you mean in the table? Honestly, I don't know that it matters. I mean, that whole table thing is called a confidence interval. So, I mean, technically, if they wanted you to know, we use a specific number, they would probably state it. We typically use the 0.05 when we're doing it, but I mean, it's a statistical thing where they're like, I am 95% confident, or 0.01 be like, I'm 99% confident, right? So I suppose if they asked, you could do either one. Number 1. Okay. Okay, hold on a second. Let me go back to the other questions, and I will get to the table in a second. I have to pull it up too, so. Okay, so Sonia asked for briefly, coupled reactions. I mean, it's really just the idea that you have some reactions that use ATP and others that make ATP, or rather some that use energy and make energy, right? So if you have a reaction that is essentially endergonic, right, I would have to put energy into it. it wouldn't happen by itself. But if I couple it with an exergonic reaction where it's releasing energy, then the other reaction will now happen because that one released energy, and now it's using the released energy to power the endergonic reaction. In the end, that's just the idea of coupled reactions. And you have a lot of those, well, in a lot of different places, right? But one place you will see that is like cellular respiration, where there's one reaction that essentially provides the energy for another one to happen down the line. Okay, let's see. Okay, so yeah, let me look up the 2013 because I have it. I just have to pull it up on my computer. That one I actually have, I think, the PDF or something. So give me a second. But yes, effectively the... Sorry, I have many, many different copies of things here. A lot of 2012s for some reason. Okay, give me a second. Still trying to find it. Yeah, so I mean, in this case, your null hypothesis is that putting, what is it, the right banana isn't going to make a difference. So yes, Audrey's right that you would expect it would be like even distribution of flies, across all the different areas, right? And then observed would be whatever data they gave you, which, sorry, I'm having some trouble finding right now, so I'm still looking. Why is it not popping up anywhere? Okay. Okay, let me try a different method here since my stuff is not working. Let's see. My computer is very finicky these days. 2013 VP. Okay. Oh, thank you, Audrey. Don't know why mine was not pulling up, but this helps because then I can just click on it and there it is. Okay. So yeah, the idea is, yeah, sorry, not banana. I don't know why I thought it was banana. But yeah, glucose soaked cotton versus dry cotton. And the assumption would be that the null hypothesis is the glucose soaked cotton won't make a difference, right? And so when you have the table here, right? Oh no, that is a banana thing. So yeah, the observed would be what you see after 10 minutes, right? That you have essentially, what is it? The middle, and yeah. So you would assume that if you would add up the number of flies that you have here, which is, yes, 60. So the expected would be that 20, 20, 20, right? That you would have even distribution. Observed is what you see, and then they want you to calculate the chi-squared for that. Okay. Okay. Any other question? Oh, Manish, taxonomy. Okay. How detailed do I think we would need to know taxonomy stuff? Like, no, I mean, they have totally moved away from having you memorize those types of things. I would say from a taxonomy standpoint, they want to know more like the question that Shweta asked about building a cladogram, right? I can see them asking you more about like reading a cladogram or building one from evidence. I think they're not likely, they don't really seem to expect these days for you to like memorize those things. So I feel like to a large extent, what they're going to want instead is, can you read one? Do you understand what they mean? Can you, use evidence to build one? But I don't think they're actually going to make you memorize it. So definitely not. I mean, like a lot of that terminology, like we never talked about Tudor stones, and there's a reason we didn't, because the AP exam doesn't really need you to know them anymore. So no, I don't think, I would not spend your valuable time memorizing cladograms. I doubt very much that that would give you much of an advantage. Okay, let's see. Okay. Yeah, I mean, I think that question it's tricky because you want to overthink it. It's very normal to. So just remember that. Also, I guess another contextual question is like, granted for you guys, they're allowing like any kind of calculator, but back in 2013, they pretty much said like you can only use a four function calculator. It can only add, divide, subtract, multiply, and do a square root. Like that's all they wanted people to do, right? So in that sense, like also the math they would expect you to do was going to be pretty simple given that. And also remember, they don't typically want you to know, you know, a lot of math per se, right? I mean, they want you to know math, but they want you to look more at like the equations. They give you like 2 pages of equations. So, and don't be afraid to use those. Like they don't expect you to do that. Okay, so Shweta, how in depth? Same thing, like I wouldn't stress the details. I mean, yeah, you should know. The only time I'd say you want to know details is if they ask you for an example and it now gives you something to give them an example of, right? But they have, so this is for the AP, not the, what is it called? SAT2. The SAT2 sometimes does like stress certain details and you never know which ones it's going to be. But for the AP exam, they've really moved away from that. They're really moving, what they really want to know is like, do you know general concepts? And then can you now apply them to these questions that they ask or data that they show you? But so I wouldn't actually stress about little details. Don't study all the details. I mean, understand the big picture things, right? Like I would understand the general idea of how um, cellular respiration works. I would understand the general idea of like how enzymes and proteins are, like things like that. Evolution, I'm sure, is probably a big topic for them, and being able to connect evolution to things like proteins and DNA and things like that. But at the end of the day, they're not looking for you to memorize a ton of stuff. Like they've really moved away a lot from that. General tips, I think you have to, well, when you're reading the multiple choice, really kind of get in their mindset, I guess, and really try to think about like, why would this be wrong? Like that's kind of the best way to go about it. And hopefully, if you've looked, it sounds like you guys have looked at quite a bit of, a good number of tests questions right now, right? You're going to start to sort of notice how they ask a question or what makes something wrong. I will say that with the AP exam for the multiple choice, they tend to have like the right answer. And then there's always an answer that like sounds pretty good, but isn't quite right. So you really have to be a little bit more, I guess, savvy, for lack of a better word, right? You want to think about like, okay, is this really right? Or if it seems a little too, something about it doesn't seem right, then don't be afraid to like say, okay, that's probably not the right one. Get enough sleep. I know you guys are stressed, but honestly, like if your brains are tired, a big part of this test is your ability to think critically. And so if you're tired, you're not going to be able to do that, right? I don't think the textbook questions hurt, but I don't think they're like the AP, no. I mean, generally speaking, right? I mean, it is the books that they know that those are the books that students are looking at, but I wouldn't say that the questions from your book are necessarily going to be any sort of indication of the AP exam. I don't think you should worry too much about scientists. I mean, I don't think it hurts to know their names, but they're really not looking for you to memorize and spew back like, oh, this guy did this and this guy just delivered, discovered that. I think that if anything, they could put a question in the context of what someone may have discovered, and it certainly then doesn't hurt for you to know how they did it. But that's kind of more about like experimental design. Like I would say one thing that students struggle with is the idea of like reading data, interpreting data. That's okay, but like experimental design, like designing an experiment sometimes tends to be the hardest thing for them. And same thing like lab concepts. I don't think it hurts to know the general idea of a lab concept, but they're not going to quiz you about exactly how this lab concept works. It would probably be just in the context of another question, but it wouldn't be like something really, detailed. Yeah. Any other questions? I think you have to remember, generally speaking. Oh, well, for free response, sorry, because I talked about multiple choice. For free response, remember that, first of all, each question, like each section of a question, is not worth very many points. So, what you have to do in its essence is you have to say enough to answer the question and answer it well. Don't be afraid to give examples, don't be afraid to define things. But at the same time, you actually want to keep yourself from spending too much time on any given question, right? Because if part A is worth a maximum of 4 points, let's say out of 10, like you don't want to spend all your time on that, because then you're going to end up with a situation where you've written lots and lots for this one question, but maybe not as much for another question. I do think sometimes people run short on time because of that, right? So it is a trick to kind of give them enough information to give you points for. So that being said, like don't leave anything blank, and when in doubt, write a little more than you think you should. But you also don't have to write this huge essay for like a short question, because they're really only expecting like a half page, right? So in that sense, also be conscious of like, what do you need to say, and make sure you say that. Like if you give it some extra detail, that's fine, but make sure your main point is there, because it could be that you write something very detailed and descriptive, but it doesn't ultimately get at what gets you that point, and now all of that time is sort of spent on something that maybe you could have spent something, more time on something else. Lab and experiment design questions. I think in those questions you want to be detailed. What I mean by that is not like a step-by-step procedure, but I can see how if they're asking you to design a lab experiment, I don't know, for like photosynthesis, and you're like too vague about what you're trying to do or how you're trying to study it, then that doesn't give them much to give you points for, right? So I think to some extent for those lab design experiment questions, like you want to show them things that you understand, like here's how I would do it. Or I think sometimes they ask you questions like I think the first question, right? I think they say like, what's one thing you could change? Like be very specific and show them that you understand things like controls and not varying too many things at once, and like only changing one variable at a time. um, being maybe a little bit more descriptive about this is how I would collect data, or like I think again, not going to the level of detail of like here's the exact procedure I would use. They're not looking for that, right? But they're looking for, can you summarize fairly completely but concisely how you would do an experiment to test something, and show them that you would have like a good experimental technique, right? Like here's how I would set it up, or here are the controls I would use, or all of that kind of thing. I think that's what they're going to be looking for, because at best it's like one section or two maybe of a question. But I don't think it would be, well, it probably wouldn't be like a whole question, I suppose, right? It seems unlikely that they would have an entire question. I can't say that for sure, because I actually don't know. But I can say that in terms of what I read from students, I can see how if you're too vague about how to do something, then it doesn't give the reader much to give you points on. I think you get to write notes. So yeah, fifth period didn't get to see it because I didn't copy in time, but I think essentially like during that first 10 minutes, like you can write stuff down on an outline. Okay, things may have changed a lot since I took the AP, because you all weren't even, alive, I think, when I took the AP. But as I remember it, and seniors feel free to correct me if I'm wrong, like you can read and you can outline, right? And so you can write on the questions, but there's like a separate little like sticker tab, and they won't let you like break the seal on the writing papers until you're ready to, right? Until they like say like, oh, your 10 minutes is up. So in that sense, like you can totally write stuff down. You're just not allowed to actually start writing your essay yet. But for the, what is it, 2014 test, they actually have like a paper where it's like, here's a page where you can take notes. We're not going to grade these notes. We're also, but you can take notes on them. So I think that essentially they'll probably do something similar like that for AP Bio. Now, yes, I mean, I would imagine the questions we did on block probably are more similar what you'll actually see on the AP exam. Because that was a 2014 test and that's after they did the redesign, right? So yeah, it's a lot of application. It's a lot of like, I do want you to understand this basic concept, like, chemiosmotic theory or something like that, right? Which has to do with the electron transport chain inside respiration. And so yes, they want you to understand it, but they also want you to apply it to things. So yes, for sure, like make sure you can kind of take their question and run with it, as opposed to they're not necessarily looking for you to regurgitate a bunch of information back. Okay, any other lingering questions? Did I get everybody's questions? I think I did, right? Okay. So I guess if that's it, we will call it a night for now. Slightly higher attendance, 8 people, woohoo. But I hope it helped. And I hope you guys will now, don't sleep too late. I know you guys are stressed, but really like get some sleep. Your brains will thank you for it. Because if you're tired, you're not going to be able to think as clearly, right? All right. I'm still available tomorrow by email if you have questions. So feel free to send them. And I'll check my email all day. I'll be running around a little bit, so I might not respond immediately. But you guys are very welcome. But like I said, send me an email. I will check email and I'll get back to you guys as soon as I can. Okay? And you're welcome, guys. Have a good night, okay? Oh, dance competition was good. Thank you for asking. Yeah, I think Gina was happy that she did the move that she was worried about. So I can let her tell you about that. Okay. I will tell her you said she'll really like that. No problem, guys. All right. I'm going to go clean my kitchen or something now. All right. I'll talk to you guys later. Send me emails. Good night. Go to sleep soon, okay? It's nearly midnight. I know you all sleep really late, but get some sleep. I'll try to tell you that, right? I can at least try to say that. All right. Bye, guys. Good night.